Okay, our next speaker is Dr. Kathy Boris Lawrie, and she's an internationally recognized scientist whose work bridges retrovirology, RNA biology, and cancer. She discovered how RNA-binding proteins like DHX9 regulate mRNA fate and viral replication, and the concept that she proposed that specialized cap-dependent translation explains how viruses and stress cells bypass normal protein synthesis. Her research revealed non-canonical RNA-protein interactions as promising therapeutic targets in cancer. She earned her PhD from George Washington University and trained with Dr. Temin as a postdoctoral fellow. She began her faculty career at The Ohio State University and later chaired the Department of Veterinary and Biomedical Sciences at the University of Minnesota. Today, her talk is going to focus on retroviruses as windows into RNA biology. Let's welcome Dr. Boris Lawrie.
Thank you. Good afternoon. I'm noticing that some people are quite cold, including Dr. Rayla. I'm wondering if we shouldn't stand up and just take a physical break. Does anybody want to do that? For Rayla. Yeah, okay. You look like you're freezing. Okay. All right. Well, I'm very privileged to be here. And in my talk today, I want to start off with a vignette about Dr. Temin and how that's led into my work ever since. And then the second part will be about some science. And so I'd like to say that, okay, here we go, the famous pointer. Okay, so I'd like to talk about Howard's sphere of influence. As you can see, Howard's up there with his family in the first sphere, his trainees, his faculty colleagues, students, and citizens. And so I was able to join this sphere starting in 1990, where 90, I talked with him, and he said, "What do you want to do for your postdoc?" And I said, "Something new." And he said, "How about chimeric retroviruses?" And I said, "Okay." And so that's what we did. And so I was then able to join that large group of people in the sphere of influence of Howard. So chimeric retroviruses. So why make chimeric retroviruses, and what are they? So chimeric retroviruses that we made were a chimera, let me see. Okay, there we go. Between bovine leukemia virus in black and spleen necrosis virus in white. And the point is that we've heard a lot about chicken retrovirus. Bird and murine, avian and murine retroviruses are genetically simple. They have a fairly straightforward, elegant life cycle. And the more complex retroviruses, we heard about HTLV, HIV, and HTLV's cousin is the bovine leukemia virus. They have additional gene features that give them a complex retroviral replication cycle. And specifically, they have regulatory genes that really supercharge the viral gene expression. So we made chimeras where we stripped out the ability of BLV to make a supercharged virus by just simply choosing spleen necrosis virus, an avian-related virus, that has a constitutive promoter, and also post-transcriptional control elements that we ultimately have studied. And we did this along the lines of figuring out basic biology, but Howard articulated a model for an approach to a preventative vaccine. And so we wanted to against HIV, we didn't want to start with HIV, we didn't want to start with HTLV, pathogens to humans. We moved to the BLV model, where we could do animal model studies. Okay. And so the animal model studies are where I got to know Čestmír Alterner. And Čestmír is shown here with his wife, Veronica, who also collaborated with us. And this sort of busy timeline just shows the dichotomy. I'm at that end of the timeline, and Čestmír started back over here, where he was interacting with Howard reading Howard's papers and became interested in his work in the 60s, and then wrote to Howard. And Howard and so Chesmer at the time was in Czechoslovakia, behind the Iron Curtain, and writing to the idea of coming to the United States was quite a heady idea. But he wrote, and Howard supported him to come, and they wrote an Eleanor Roosevelt Fellowship that was a year-long sabbatical in the lab. And Veronica and Chesmer came for their first of 3 tours of duty. Let's see, here we go, 1, 2, 3. Okay, and so the upper part of the timeline is really just to show you the sort of dynamic political situation that happened in this time frame. So the first time Chesmer came, he was behind the Iron Curtain. He went back just after the Prague Spring, when the tanks rolled into Czechoslovakia, and the Iron Boot came down harder on the dissidents in that country. He was there then told, you need to work on something more important than a chicken virus. And so he decided, I'll work on a Bratislava bovine leukemia virus and develop animal models. So in that time, here we are. There we go. There's the Nobel Prize. Howard was awarded the Nobel Prize for work on chicken or bird retrovirus. And Chesmer came back. And then fast forward, here we are. Conditions completely changed, where the Iron Curtain falls and Chesmer is able to come back and forth, no problem. It is under those circumstances that we were able to Howard put us together. So Howard put me, probably one of his last trainees, together with one of the first people that came to the lab. And this has been a really very rewarding collaboration. And it's almost remarkable, just looking back, looking back at my pictures and thinking about this history, it's remarkable that Howard supported Chesmer, of course, it's wonderful, to come to the United States. And then after that facilitated Chesmer's career for sure, and led to bovine leukemia virus work, where we could then test these novel viral constructs. So I show those novel viral constructs. They were created in a time, over the period of time that Howard wrote that article, became sick. And I would like to point out Dr. Bill Sugden here, who was an immense supporter as we went through various renditions of these little vectors into coviral vectors, and then finally the single genome vector that had one RNA that made all the viral gene products. So it was really quite a remarkable experience from that frame as well. Okay, so I would like to say that he brought us together, and we were able to, in a small way, identify his or prove his theory that a genetically simple retrovirus could be made of bovine leukemia virus that was immunogenic, non-pathogenic, and even protected against a challenge virus. So I'd like to say that Howard didn't teach science. He didn't only teach us science. He taught us how to work together and how to help ideas thrive as we all left the lab. So I would like to point out that I think that is a tremendously enduring legacy, and I'm totally privileged to have been a part of it. And very proud for you. So now I'm going to slip into some science where, really by making these chimeric retroviruses, we were taking RNA from a simple retrovirus and connecting it to a more complex retrovirus. And so there were a lot of activities that we had to make sure were maintained, and even some that we didn't understand. And so ultimately, by doing that and following the little clues that were breadcrumbs, we've identified a novel translation control pathway. And so I'm just going to, it has to do with the RNA cap. So I'm going to start off in studies of HIV, just a brief, we already heard about RNA, so I don't have to spend much time there. And then I'll talk about specialized translation, which means non-canonical translation, and then how that enables the virus as well as cells to thrive under stress. So we heard a lot about the transcription start site from Doctor Who, here we go. Here's the RNA. Here's the guanosine molecule. I'm not counting G's. There's a G. And it turns out that if that RNA is not protected at the 5' end, that 3' that phosphate group will be a fodder for DNA degradation enzymes. So the RNA absolutely has to be protected from decay, and it's going to become epigenetically modified. In other words, it's going to get another molecule stuck on the 5' end. And so instead of one phosphate there, you can now see that there are 3, because another G molecule was added in a reverse orientation. We always hear about DNA, RNA, 5' to 3'. Well, this is a case where RNA is linked 5' to 5', not by genetic encoding, but by epigenetics. And that protects the 5' end of the RNA from decay, and it also gives the RNA a shape where it has aromatic rings that can interact through sandwiches with very specific RNA cap binding proteins. And those proteins are needed throughout the whole life of the RNA to get spliced, to qualify for export, and for translation. So one of the proteins that can interact with the 7-methylguanosine cap is, where's my pointer? Oh, it's not on here. Next slide. Is TGS1, trimethylguanosine synthase. So this comes in and can interact with the cap just like all the other M7G binding proteins, and it leads to 2 more star-labeled methyl groups being added to this molecule. And those simple, small but mighty methyl groups change the structure of the cap and its ability to interact with RNA and protein partners. Okay? So just for instance, it disqualifies those traditional cap binding proteins from binding. So you lose one thing, you gain something else. We've just identified something else. And secondly, it changes its ability to do Watson-Crick base pairing. So you can see down here, biochemistry students, and then the blockage of the methyl group blocking the ability to form base pairs. So that's very important, we believe, to the structure of the RNA. And so how important is this? And I think I forgot to mention, this trimethylguanosine cap, TMG, I'm going to say TMG a lot, sorry, TMG, was really well known for some small non-coding RNAs, such as have been studied here at Wisconsin. And for years, and very seminal studies in the nuclear export, or the trafficking of those RNAs were done here in the Dahlberg group. So it was really surprising when T. Zhang found that HIV had TMG-capped RNAs. All the other ones were non-coding RNAs. Why would HIV have this? Well, it's going to change its protein partners, obviously, but does it matter to the virus replication cycle? And so the way this is first done, we did this, and T originally did TGS1 knockdown, and what we find is that we get a 10-fold reduction in the amount of the GAG protein that Lou showed beautiful pictures of. And we also get, of the viruses that are left, we get a tremendous reduction, 3-fold magnitude in the infectivity of those viruses. And that is due to the lack of it's attributable to the elimination of the TMG cap. So what I'm going to tell you about in the rest of my talk is a cartoon, and I'm going to show a sprinkling of evidence, not complete, but sprinkling to give you an idea. The top pathway is the canonical pathway, where EIF4E interacts with this cap to facilitate ribosome recruitment. There's our purple ribosome. And this really helps the cell make lots of protein, but of course cells shouldn't make lots of protein all the time. They do often upregulate this pathway in cancer, in cell transformation, but it's really important to modulate the amount of protein synthesis. And in fact, the cell cycle during G2M inactivates the important kinase that leads to the phosphorylation to control and dysregulate EIF4E. So this is highly regulated, both physiologically, can be affected in disease. Okay, and in our work, what we're finding is TMG cap no longer fits. It's red, and it's a different shape, and it interacts with a different set of nuclear proteins that had not been previously identified to interact with the TMG cap. Syreportin was what was originally identified as the single protein that I believe was interacting with the TMG cap. And that leads to specialized translation. Specialized, it's kind of a shaggy word, I guess you'd say, John. It's what it means is that it's used in the field to say translation that is independent of the normal traditional mechanism. So how did we figure this out? Okay. So this is a smattering of data from some papers over the years. And here again is our cap and being inhibited by mTOR. When the factors are together, they go from being called E to F. It's not a typo. And when we look over here, can you still hear me? Yeah. When you look over, Okay, there we go. The first lane is uninfected cells. The second lane is uninfected cells. There's a typo there. When we add an mTOR inhibitor, what we can see is that the cell translation goes way down. The third lane is the HIV-infected lane. HIV reduces total cellular translation. And when we add the catalytic mTOR inhibitor, a very site-specific active inhibitor, it goes down even further. But when we look below, we do metabolic labeling by various mechanisms. And what you can see in that IP of GAG, of cells that were labeled with puromycin, and we see that by Western, there's no change in the amount of GAG protein. We've done this in several different types of assays. Whereas a host gene is downregulated. And if you look at the Western blot down below, what you can see again, minus and plus the mTOR inhibitor, that we see a reduction in GAPDH, TAT, REV, and NEF, which are made by the multiply spliced RNAs early in the viral replication cycle, whereas GAG and GAG-POL, I'll show you in a minute, and envelope are not downregulated. And this can be shown by several different types of experiments with metabolic labeling. So we said, well, why is the HIV structural protein not downregulated? So we looked across infection, and what we see is in the progression of infection in hours, that the 4E repressor gets activated after the virus is replicated. But if we delete or put in a stop codon to prevent the expression of some of the accessory proteins, such as VPR and VIF, we do not see that reduction. And so we took and we just can compare the VPR protein to mTOR treatment, and what we can see is that VPR is sufficient to reduce the polysome profile of the of cells. So that led us to think, well, VPR, we knew from Michael's work, was causing cell cycle arrest, and that was really what facilitated this work. We said, how could the virus be making GAG in our experiments when VPR was causing cell cycle arrest? Because of the activation of the mTOR inhibitor. So we went to work to figure out what were the proteins that were bound to the HIV unspliced RNA that makes the GAG and also the singly spliced that makes the envelope protein. What were the proteins that were bound? And we looked at the typical EIF4E, and this is an immunoprecipitation from the polysomes, and on this blue line, of highly translated RNA. And we can see 4E is there, 4G, HIV, a little bit of unspliced, but we see the multiply spliced. We see the GAPDH. But when we add taurin, the inhibitor, this complex is gone. This is gone, this is down, GAPDH is down. And then when we look at a nuclear cap binding protein that we knew from other studies interacted with HIV, we could see that more of the unspliced RNA was in this complex, and it was not reduced by taurin. So in other words, the translation continued under inhibition of mTOR. We tied this together with some other experiments that we had published on that RNA helicase A, a dead box helicase, is important for translation of several different retroviruses, and this came out of work with the chimeric retroviruses. And so we knew RHA was important, and this is a complicated experiment that shows that if we knock down RHA, we get a reduction in translation of the unspliced RNA, but the multiply spliced RNA doesn't care. If we treat with taurin, where's that? Okay. Treat with taurin, the GAPDH and the unspliced RNA go down, but the multiply spliced RNAs go down that make the regulatory proteins, whereas the structural proteins don't. So we thought that perhaps looking for the binding site of RHA would obviously play a role in the specialized translation. And so what we were able to do is show that this segment of the 5' this is a little piece of the 5' end of the virus. This PBS segment, which is used in the virus to facilitate reverse transcription, does double duty by also interacting with RNA helicase A, we showed in other studies, and it forms a three-way junction, as you can see from this SACS. We can make a single nucleotide change. Again, this RNA is so sensitive to fragile, small changes, a single nucleotide change, and the folding differs. And we go from having GAG being translated during mTOR inhibition to the virus now unable to translate during mTOR inhibition. And so we've shown that this mutant loses the TMG cap, and so that meant we had a way to study this without using a drug that might have off-target effects. And that virus was poorly replicating, but if we left it in culture, over time, the virus would come back up and gain the ability to have 5' caps and resume replication. The sequences are highly conserved, and we've shown in other strains that this is conserved. We then realized that RHA was acting as a dimer, and so that has led to our newest work, where we are postulating that RHA interacts with two ends of the viral RNA to bring that together, to bring the REV response element and REV to the 5' end of the RNA to facilitate communication between the cap, which is absolutely necessary for the escape from the nucleus, as well as the translation of the RNA. And so when we look, we found this 3' element, and what we found is that it's really conserved, except for in some long-term non-progressors where there are mutations. We are now studying those mutations to test our theory. So we were able to bring this back to the patient. We hope to bring this back to the patient to understand the role in the persistence of the virus. So this is what I've shown you today, and I'm going to stop pretty soon. We've gone on to look into cancer cells. We've done genome-wide screens, and we now know that what we see in the retrovirus is conserved in the host. Makes sense, convergent adaptation by the virus of a host mechanism. And we've been studying that first in canine sarcomas, and we've been showing that there's crosstalk between the top pathway and the bottom pathway, and that the specialized pathway is necessary for cells to recover from the mTOR inhibition of the top pathway. So that has significance for controlling translation in various types of situations or in neoplastic cells. Okay, so these are my take-homes. And I'd like to thank all of the people here, and most awesomely, Dr. Temin. And I really feel that this science legacy is one of collaboration and connection. We're all here today. Many of us are even much more connected than we ever would have thought 30 years ago. And so I'd like to thank Dr. Temin and the family and our organizers for being here. Okay.
Okay, questions for Dr. Boris Lawrie? We're all waiting to see if Dr. Scherer asks the question or not.
I know what he's going to ask.
Spoilers.
Then you can just answer it. No, I'm kidding. No, I'm going to ask the same question I asked Wei Su, which is, have you guys looked at other viruses to see, how prevalent trimethylated caps are in the retroviral world? And I was just curious.
Yes, that's an obvious, really great question, Nate. And so, yes, we have. We've looked at avian sarcoma virus, and the answer was that we could not find positive data. And our positive control are the SNO RNAs, usually SNO U3 RNA. If we can pull down that, and we compare our PCR qPCR, we would have the positive control, but not the simple retrovirus. But we did the same experiment in several different types of cells infected with human T-cell leukemia virus, a complex retrovirus that has those TAX and REX genes that are so potent in the pathogen that contribute to the replication cycle. And the answer is yes, there, in several different types of cell types. So, yes, I would really like to look in patients. And these experiments require quite a bit of RNA, so it would have to be a pooled amount of patient RNA. So, yeah.
Is that the question you expected?
Excuse me?
Is that the question you expected?
No, it wasn't. But it was better, way better. Yeah. Okay. All right. Thank you.
Dr. Lambert has a question.
Oh.
Yeah, that's a great talk. Fascinating. So TMJ, you mentioned in your slide, is also found to be upregulated in certain cancers. And what cancers are they? Are they a specific type of cancer? And I'm just wondering if any of them are virally associated.
So first of all, I will be, that is my goal right now in life, really, is to figure out what is the role of this in disease. And we did work in osteosarcoma. We don't think it's necessarily that, so the CAP, we think there's upregulation of some of the factors that facilitate the ability to make the TMG cap and do specialized translation. We don't know if that's the whole reason that this contributes, but we know if we can knock down specialized translation, the cells no longer grow in soft agar or these types of ex vivo assays. So we've looked in hemangiosarcoma, osteosarcoma, and we're doing work now in human ovarian cancers. And so that's as far as we've gotten. But DHX9 RHA is upregulated in many different types of cancers, including lung. And it is on a chromosome locus that's a cancer locus, 1q25. And so it's possible that the upregulation of that gene leads to upregulation of this pathway in some cancers. Yeah.
Fascinating.
Yeah.
Thank you for your talk. So interesting, and I was very fascinated, of course, by the first part of your presentation and the connections with Chesmere Altener. And I was wondering, was Jan Svoboda in one of your photographs?
He was.
Yeah, because Jan Svoboda was a very major and very close colleague of Howard's, who came and worked in his lab, and Howard made 2 trips to Czechoslovakia when it was behind the Iron Curtain in the 1980s.
I did not know that.
I went with him, and that was a very, very major. And he was at that 1993 meeting.
Yes.
No, in 1993 when they, after Howard became ill, when they had the book, and Jan Svoboda has an inscription in there. So I was just wondering if he was in your photograph, but
He
I
was.
guess, yeah.
He
I don't
was,
know if
and
you
he-
met him too.
Well, I met him through Česmír.
Yeah.
By going to Slovakia. By the time I went there, it was now Czech. It was Czech Republic
Yeah.
and Slovak Republic, and we interacted with Jan, who was in Prague, and Česmír at the Czech Center for Cancer, and then we would go to the Slovak Cancer Institute,
Yeah.
and so they interacted. But we got together at meetings, and all of the people in that little photo were at a meeting in the High Tatras Mountains of Slovakia at an anniversary of that Cancer Institute in Slovakia. And Jan was there, and other people that were studying bovine leukemia virus. Lou also studied bovine leukemia virus. So, yes, we interacted in that way, but Jan wasn't part of the work.
But when we went, it was very chilling. We had to be very careful of what we said at any point, and it was a very frightening experience. But they were very close friends.
That's really interesting.
Okay, let's thank Dr. Boris Lawrie one more time.